Hi, everyone. Welcome to a podcast on biotechnology. In its simplest form, biotech is using cellular processes to make a product or a procedure that's beneficial to us. So, we're just going to kind of touch on a few here, but it's a fascinating topic. So, you can look back through your notes or you can even Google some of these and find out more about them. Okay, the first thing we're going to talk about is cloning, and specifically reproductive cloning. And you can't really talk about reproductive cloning without talking about Dolly. Dolly was born in 1996, and the reason she's such a big deal is this is the first mammal that we successfully cloned through reproductive cloning. And she lived about 7 years and she died of a disease common to sheep, little baby sheep. So, how do we do reproductive cloning? Well, basically, what you're going to do is you're going to get an egg cell from just a random female sheep, and you're going to, using a needle, suck out the egg, its nucleus. So it has no nucleus at all. You just kind of have this empty egg cell. Then what they did is from another female sheep, they got a cell and they isolated the nucleus and they put a nucleus into this empty egg cell. All right, so this nucleus came from an adult sheep and was just put into this egg cell. And then the egg cell was encouraged to divide and made a blastocyst. And then that was put into a sheep. That sheep became pregnant, and that was Dolly's surrogate mother. So Dolly was an identical clone to the sheep whose nucleus they took. So why are we using so many different sheep? We have one sheep where we got the egg cell from, one sheep where we got the nucleus from, another to be the surrogate, was to make sure the one that they got the nucleus from, that her DNA was identical to Dolly's DNA. And it was. It was a successful clone. And that's how we do reproductive cloning. Reproductive cloning could even allow us to bring back some extinct animals. So this is a baby woolly mammoth. It was found in a Siberian body of water that obviously was frozen. And through reproductive cloning, they might be able to piece together enough DNA and whatever they can't get out of this woolly mammoth, they'll use an Asian elephant and create a brand new woolly mammoth. So we could actually bring back extinct species. Therapeutic cloning is a little bit different from reproductive cloning. Here we're not cloning an entire organism, we're cloning just cells. So what's usually used is embryonic stem cells, although recent work has allowed adult stem cells to be used and kind of recall the pluripotency of embryonic stem cells. And we culture, which means we create many of these embryonic stem cells. And then we can expose them to different cell signals or hormones to get them to differentiate into the cells that are needed. Blood cells, nerve cells, or heart or muscle cell. What if we don't need an entire cell even? What if we just need a gene? Then we're going to use gene cloning. So what we do is we pull DNA out of a human cell, and then we target the specific gene we need. This example is growth hormone. Insulin is another very common example, and we will cut just that gene. So this is our human gene for growth hormone. Then we're going to get a bacteria cell, and we're going to pull out a plasmid. We're going to cut open that plasmid, and then we're going to introduce the human gene and make what we call a recombinant plasmid. We call it recombinant plasmid because it has bacterial DNA and it has human DNA. So whenever you have recombinant DNA, that's DNA from two different sources. Okay, well, plasmid's just a circular piece of DNA. It doesn't have any equipment or machinery to make anything. So then we need to get this back into a bacteria cell, and we will rely on transformation for that to happen. And once it's back in the bacteria cell, it will have the equipment it needs to make the protein. And sure enough, this plasmid with that human gene will make the protein that we want. In this case, human growth hormone. But as I said, this is actually how insulin is made for a diabetic. So how do we cut that human DNA to cut around that gene we want and the plasmid? We use something called restriction enzymes. Restriction enzymes occur naturally in bacteria. They kind of act like an immune system for bacteria. They will chop up any foreign DNA. So, if a virus injects DNA, a restriction enzyme could get in there and cut that DNA before it even became an issue. But we use them. So what we do is we use them to cut DNA. In other words, restriction enzymes are scissors for DNA. And we can make two very different cuts. We can make one that will result in what's called sticky ends. So what would that be? That's when it cuts and it leaves a single strand of DNA, and DNA does not like to be single-stranded. So what that DNA will do is find complementary DNA and bind to it. So these are called sticky ends. It can also make a blunt cut, and a blunt cut is simply when it just cuts DNA so that there is no single strand. And we usually use those just if we're breaking apart DNA for like DNA fingerprinting, like what you'd see on a crime show or something like that. We use sticky ends if we're trying to clone a gene. So we would cut the insulin gene as well as the plasmid so that it would have a sticky end to it. Once we cut DNA with a restriction enzyme, we call those chunks of DNA restriction fragments. Restriction enzymes are very, very specific about where they cut. They recognize base sequences and cut there. And these sites where they cut are called restriction sites. And that's how we cut DNA. We don't just use restriction enzymes in this idea of taking a gene from one organism and putting it into a plasmid just to clone genes. We also do this for creating genetically modified organisms, or GMOs, which I'm sure you've heard about GMOs. So a genetically modified organism contains one or more genes introduced by artificial means, usually from like a completely different organism. If it's transgenic, then the gene even came from a different species. So very similar, we'll get the gene that we want, we'll get a plasmid, we'll shove the gene into the plasmid at the restriction sites, we'll cut the plasmid with a restriction enzyme, and then we'll cut the gene with a restriction enzyme using the same restriction enzyme with sticky ends. We'll put them together, and ligase will seal up those cuts, and now we have a recombinant plasmid that we'll put into a plant cell, and then that will actually act as a vector. So a vector delivers DNA. Okay, and I'll put it into the plant. So why would we ever genetically modify anything? Well, we use it in our foods as well as sometimes like things that we use for industrial reasons like cotton. But it delays ripening, it adds nutrients, it encourages further growth. So there's a lot of reasons we create GMOs. And I know it's kind of a hot topic, but there's not a lot of research that says that they are bad for you. Although there is a lot of work done on creating genetically modified organisms that can survive a herbicide or a pesticide like Roundup, that allows so much of that herbicide or pesticide on your food that can be bad for you. So it's definitely an interesting topic. Now, another reason we might use restriction enzymes is to create DNA fingerprints or a DNA profile. So what we can do is we can take DNA and we can introduce a restriction enzyme that will cut the DNA into chunks or fragments. We'll then run a gel electrophoresis, which I'll explain in a second, which basically means the DNA is run through a gel, creating these bands. And then we can use that to match to other bands. So in this kind of silly example, I have blood found at a crime scene, and then I have two suspects. So what I would want to do is I'd want to see, okay, whose DNA matches the one found at the crime scene. So this is my crime scene DNA. Obviously, it's not suspect 1, suspects 2 DNA matches. And that's basically how we'll use a DNA fingerprint. Okay, so what's gel electrophoresis? It's a neat technology. So this is agarose gel right in here. And agarose gel is made from seaweed. And it's a lot like Jello, that same kind of consistency. And basically it creates a matrix. So imagine if like you had a very, very dense forest, an extremely dense forest, and you wanted an elephant to run through it as fast as it could. Well, the elephant couldn't because it would have to stop and slow down and move between the trees. But if you asked a mouse to run through it as fast as it could, it really could. It wouldn't be hindered by those trees. It could run as fast as it can. All right, so why am I giving this example? The agarose gel is kind of like your forest, and the chunks of DNA is your elephant, your mouse. The really big molecules can't move very quickly, so they're stuck up here. And the little molecules can move very quickly, and they're all the way down here. So agarose gels sort DNA by size, with the bigger ones up towards the top and the little guys all the way at the bottom. So you put your DNA into these wells. How do I encourage my DNA to move? Well, you guys know that DNA is negatively charged. So what we do is we attach electrodes. We put negative up here and we put positive down here. And the negatively charged DNA will be repelled by this negative charge and attracted to the positive. So the DNA will move down. And the bigger molecules will just move a lot slower. And the little molecules will move a lot faster. And then that's how we get these bands. Last but not least, we're going to talk about PCR. Okay, PCR stands for polymerase chain reaction, and this was huge in biotechnology. And why this was such a big deal is using a machine, and it looks like this right down here, kind of like a chemical-looking shoebox thing. We can amplify one little chunk of DNA without a cell, and we can take it from just one copy to literally billions if we wanted to in just hours. No cell needed. Okay, so how do we do this and what do we need? Well, the first thing we're going to need is the piece of DNA we want, the chunk of DNA we're trying to amplify from just one copy to literally billions of copies. Then we need tons and tons and tons of nucleotides. And then we need primer sequences because remember, polymerase cannot attach unless it has a primer. So we'll have to know some sequences of the DNA because we'll have to have a primer. We have to have two primers for this strand up here and this strand down here because remember, DNA is complementary. So we'll have two primers. And then we need a very special polymerase. And the polymerase we're going to use is actually called Taq polymerase. And Taq polymerase comes from a bacteria that lives in very extreme conditions in like hot springs. So this polymerase will not denature when we raise and lower the temperature. And the reason we're going to raise and lower the temperature is DNA is double-stranded, and it's hydrogen bonds that hold those two strands together. Well, you guys know hydrogen bonds are pretty weak. So if we raise the temperature, what we will essentially do is break those hydrogen bonds so that the DNA is single-stranded, and then we can copy it. So we needed Taq because it wouldn't denature when we raise the temperature. Okay, so very quickly, this is how we do PCR. We have our gene of interest. Okay, we raise the temperature to about 95, 97 degrees. That separates our DNA, all right? And then we inundate it with primers, okay? We're just going to put so many primers in that the DNA won't have a chance just to re-anneal. It's going to anneal to the primers. But in order to allow that to happen, we have to lower the temperature to about 68 degrees Celsius, okay? And once the primers are attached, then we can go ahead and allow the DNA to be replicated. DNA polymerase will come in and replicate that DNA, and then we'll do it again. We'll raise the temperature, separate the strands, allow the primers to anneal, and then allow elongation. And we keep doing this until we have the amount of DNA we need. And that's a really short presentation on biotechnology. I hope that was useful for you guys.